Let me start by thanking the organizers for the invitation to come to this meeting. It's really, a real honor for me to participate. And, the PDB has been incredibly central to the work that we've done over the years in computational protein design. So I don't have a lot of time, so I will get right into it. What I'll try to do in the next 20 minutes or so is give a very, very brief outline of computational protein design, and then talk about some of our recent work on modified antibody drug conjugates, something that we're calling CDR Extended Antibodies. So I'll talk about what the concept is, and then talk about a relatively simple test case that demonstrates the approach. Okay, so computational protein design. So efforts in this area date back to the early 90s and possibly even earlier than that. They're really, can be separated into sort of methods and applications. The methods rely on having some sense of the protein backbone. And of course, all the early work relied on backbones that were derived from the PDB structure data bank. The calculations utilize rotamer libraries, which again were to represent the side chain conformations. Again, these were derived from the PDB structures. We compute energies in the system using atomic-based force fields, similar to the force fields that are used for molecular mechanics and molecular dynamics simulations. And then, of course, there's a combinatorial problem that needs to be solved, and we use various techniques in computer science applied math to solve the combinatorial optimization problem. And in some instances, there's an issue of negative design because it's not always sufficient to design for what you want to achieve. You also have to design against alternative solutions, for example, like aggregation. So these methods have been used to examine a variety of things, ranging from, looking at the relationships between sequence structure, stability, and function, all the way to using the approaches for designing biologics that can be used for human therapeutics. So it's been almost 25 years since our first major result in this area. So back in 1997, Basil Dayat and I published the first fully computational designed protein sequence that actually was shown to fold to its target structure. So the image is just showing a comparison between the target fold, which I believe is the red one, it was a zinc finger fold, and the NMR structure of the designed protein. This was our first PDB deposit. It has the PDB code 1FSD for full sequence design. And to the extent that the force fields are relatively accurate and that the optimizations are working well, you can imagine that you can use these approaches to stabilize proteins. And we demonstrated that in 1998 by using the computational approaches to generate a hyperthermostabilized stabilized variant of protein G, which made it to this, what I think is a really nice cover of Nature Structural Biology. And of course, the protein that we use as a template is structure was determined by Angela Gronenberg, who we're going to hear from later. We also saw the structure of the variants using NMR again, and that PDB code is 1GB4. Okay, so CDR Extended Antibodies. So what are we after here? We're probably all familiar with the concept of antibody drug conjugates. So this is a protein that has a small molecule drug attached to it. And the antibody component of the ADC is used to target the small molecule drug to the appropriate location, usually cancer cell. And then the small molecule can be dissociated, and its activity then will be directed toward an independent molecule. And so the important features are that there are two targets. The antibody has a target, which is used for localization, and then the small molecule drug has a separate target. So there are two independent targets with two different binding sites. So there's no binding synergy between in the system. Chemibodies, which I'll show a diagram of in a moment, is an advance on ADCs that was developed by Amgen very recently. And in this case, there is binding synergy between the components of the system. So the antibody binds the target, and the small molecule also binds to the same target. So in a Chemibody system, there's one target and two binding sites. And I'll show a diagram in a moment. And what we're after is something that is an advance on top of the Chemibodies, which is what we're calling CDR Extended Antibodies. And as you might imagine, given the name, the small molecule is actually covalently attached to one of the CDR residues and is involved directly in the antibody-antigen binding interface. So in this case, there's one target and one binding site. And the objective is to achieve a high degree of synergy between the components in the system. So on the left is work out of Amgen published in 2018 that shows what they call a Chemibody. And so it's an antibody that has a covalently attached small molecule, stigoliptin in this case, binding to DPP4. And so you get binding synergy, but although it's hard to tell in this view, there's no synergy, there's no direct interaction. Sorry, the antibody is binding on a part of the protein, the antigen, that is distal from where the small molecule binds. And of course, the linker, which is relatively long in this case, connects the two molecules together. For a CDR extended antibody, we're interested in having the small molecule covalently attached to one of the CDR loops and have it directly involved in the interface between the antibody and its target. And we believe that this will provide for significantly enhanced target specificity, enhanced binding synergy of the components, and can be used potentially to, for example, rescue failed drugs. So small molecule drugs that might, for example, have off-target binding. In this format, the off-target binding can be defeated because the antibody would bring significant additional binding specificity to the small molecule. So we're quite excited about the prospect of this modality being used for novel purposes. Okay, so we wanted to test the system. And so we selected a relatively straightforward target, a monomeric version of streptavidin. As we all know, streptavidin is naturally a tetramer. The monomeric version of streptavidin, MSA in this particular case, binds to biotin with reduced binding affinity. The small molecule, of course, then will be biotin, and we'll couple the biotin to our antibody using C2 maleimide chemistry coupling to cysteine. And then for this test case, the antibody will just be a nanobody. Okay, so the approach is really divided into three parts. First is the need to identify a appropriate antibody streptavidin pair. So the antibody antigen target needs to be specified. Then we need to specify a position on one of the CDR loops to introduce a cysteine that will then allow coupling of the biotin. And of course, that coupling will require that the geometry be appropriate so that the small molecule biotin can actually still bind to the target streptavidin in a manner that doesn't disrupt the interaction of the nanobody also binding to the streptavidin target. And then, of course, it'll likely be necessary to optimize that protein-protein interface and possibly also optimizations in the framework region of the antibody. Okay, so for this test case, we selected 154 nanobody antigen structures from the PDB. The nanobody components of those complexes are shown here, these 3 different orientations of the same assembly of 154 nanobodies. And you can see the framework in gray, and then the various CDR loops from those nanobodies. CDR1 is shown in red, CDR2 in yellow, and CDR3 in blue. And then for these 154 antigen nanobody complexes, we characterize the interfaces in the systems, in the complexes, by separation distance, buried interface area, and then a shape complementarity score. Okay, so then with this library of 154 nanobody structures, we performed docking against monomeric streptavidin and generated 2,310 dock poses, which were then filtered by the following criteria to select the initial design target, the initial design scaffold. So the filtering criteria are that we wanted to select a complex that matched the sort of the average interface statistics that we saw from our nanobody structure database. Importantly, obviously, the complex, the positioning of the nanobody on streptavidin needs to block the biotin binding site so that we can have a relatively short linker between the biotin and the nanobody. And then ideally, it would be desirable to have a binding mode of the predicted nanobody interaction with the streptavidin that looks structurally similar to the nanobody's binding to its native or cognate antigen from the crystal structure. So that we would expect that there wouldn't necessarily need to be significant loop relaxation or reorientation of the complex. Okay, so applying these 3 criteria led to 7 putative complexes that we could use as a scaffold. And you can see, so for the streptavidin shown on the bottom in green, and the various nanobodies are shown in cyan, docking to the top, and you might be able to see the biotin structure, small molecule structure in its binding site. From this filtered set, we selected the B chain of protein 4NBX as the design model. And you can see it makes what looks visually like a really nice complex with between the 2 components. Okay, so then what do we do to get the biotin in? This is also done computationally, selecting the position on the CDR loop. So here's our small molecule, biotin C2 maleimide, and we're going to use coupling chemistry to the cysteine to join to make the covalent attachment with the nanobody. Okay, so the in this process, the biotin is anchored in its crystallographic position in streptavidin. And so, of course, the part of the biotin that's sort of not natural, not part of what we think of as biotin, needs to be positioned so that we can figure out what position on the CDR loop will best accommodate this linkage. So we, in the calculation, we use a polyalanine CDRs, and then we impose rotamer flexibility in the biotin tail that allows the position of this sort of C-alpha residue or amino acid, the position of the C-alpha atom to, explore the space around the system, consistent with the kind of rotamers you would expect for this kind of linkage. Then we evaluate steric clashes with the streptavidin and the nanobody, and then look for the best match for the C-alpha position. And that resulted in this complex. and you can see that the nanobody with its relatively long C3 CDR accommodates the, so you can see the cysteine atom, the sulfur hidden behind here. It accommodates the linkage quite well without any steric interactions or strain in the small molecule. And this desire to have a match between the sort of native binding of the nanobody to its antigen target and what we see in our model is actually demonstrated here. So in the crystallographic structure of the nanobody from which we derive that structure, nanobody, there are two critical interactions between two side chains, a tyrosine and an arginine on the nanobody, and side chain backbone on the, here's the original crystal structure on the antigen target. Those interactions are preserved in our design model, which gives us some confidence that the positioning of the nanobody is unstrained and looks like it might accommodate the kinds of interactions that we're trying to develop in the system. Okay, so experimental validation. We're using surface plasma resonance, SPR, to measure the binding in the system. So the wild type streptavidin, monomeric streptavidin, binds to biotin, immobilized on a chip with a KD of 7 nanomolar, which is consistent with previously reported values. We're also using a point mutation of monomeric streptavidin, serine 27 to alanine, in order to reduce the binding affinity to biotin. So we have more room to play with in terms of measuring affinity improvement. So the base system point mutation binds with roughly 1 micromolar binding affinity. Okay, so then wild type 4NBX does not bind to streptavidin at all, as measured by SPR. And then if we look at our sort of our base design, which has no optimization in the interface, it just positions the biotin on the nanobody, and then we get for free the 2 interactions that I mentioned previously that come along for the ride. So we go from a 7 nanomolar binding of biotin by itself to roughly 2 nanomolar for the complex, for the CDR Extended Antibody complex. And then for the sort of binding compromised monomeric streptavidin, we go from roughly 1 micromolar down to roughly 250 nanomolar. So we're getting 4 to 5-fold improvement in our base design. So again, this is without optimization. So we can then deploy computational design on the CDR positions to enhance binding. We selected 2 variants, either both 4-fold variants of the parent. Variant 119 is the highest ranked variant by binding energy. And then variant 149 was the highest ranked variant that still had good binding energy, but also had the largest number of predicted hydrogen bonds, with the idea that this might optimize the off rate. Here are some of the models of the interactions. I won't go through those for the sake of time. Experimentally, they actually do in fact improve the binding. So if we look at V119 binding against mobilized wild type streptavidin, we reduced the KD down to roughly 1 nanomolar. Variant 149, which was designed to have off-rate optimization, has again significantly reduced binding, improved binding down to 10th nanomolar. And as you can see from the SPR experiments, the off rate does appear to be slower compared to the parent. So we're getting an additional 2 to 15-fold enhancement of binding, including improvement of the off rate as desired. Unfortunately, variant 149, the best variant, shows poor behavior by side exclusion chromatography. Variant 119 shows pretty clean single peak. This is in triplicate, whereas variant 149 shows significant aggregation. And so in order to try to address the aggregation, we did a further optimization of the CDRs using 149 as a parent and introduced 6 additional mutations that improved the binding affinity by SBR, but significantly degraded the behavior on size exclusion chromatography. So in order to address this, and I'm coming close to the end, we analyzed our design model by MD simulation to look at what was happening in the complex. So I'm showing here the nanobody in cyan and the streptavidin sort of in brown. The biotin linkage is in there, but we're not showing it in this video. And what you can see is that this loop actually partially unfolds. And if there were different orientation, you would see that's actually pulling away from the framework region. If we analyze the MD simulation, we notice that there are sort of 2 regions that could be optimized that would improve this behavior. So one is mutating an alanine in the framework region, the valine, to improve the hydrophobic packing in that region. And the second is mutating a phenylalanine to tyrosine in order to introduce a hydrogen bond between the framework and the CDR3 loop. Simulation of this double mutant actually does show improvement, and I won't go through those details, but these curves that analyze the this partial unfolding shift to the left, which is desired. Experimentally, we actually retain the improved binding of this variant, 186 with framework mutations, and the size exclusion chromatography traces are not perfect, but they're significantly improved relative to the parent. So overall, we've been able to improve the binding by 35-fold with respect to the wild type monomeric streptavidin, and roughly 21-fold tighter compared to the 1 micromolar starting point for the point mutant. So in summary, CDR extended antibodies are a natural evolution of the antibody drug complex concept, offering sort of a new modality for thinking about new types of binding molecules. We've shown that we can actually generate these kind of proteins and improve them using computational design. And we've, in our test system, showed greater than 20-fold improvement of binding relative to the binding of the small molecule by itself. This work was done primarily by Jingzhou Wang, who's a graduate student who's going to be defending next week. Really great worker, great insight on lots of the sort of problems that we had in the system. He was aided by Aidan Aceves, who helped out on some of the MD simulation work and some of the computational modeling, as well as sort of laboratory help from Monica Bredkow, who was a lab technician. And I'll stop there, and hopefully we'll have time for some questions.
Thanks, many thanks for this very, very interesting talk and interesting technology. I think there are a number, 1 or 2 questions. Let me have a look. Yes, there is one question. Based on one of your filtering criteria, namely that the binding mode must be similar to native nanobody antigen crystal structure, do you expect the CDR loops to rigidly to rigidify upon binding, and how much variability do you notice in the CDR loops between different models? And under parentheses, assuming that the docking was not completely rigid.
Yeah, so the docking was, we did not allow a lot of relaxation in the docking. So it wasn't perfectly rigid, but it was close. And the reason that we did that was because we didn't want to get involved in loop modeling. We wanted to generate a target complex that was as close to something that was already observed as possible, so that we could essentially eliminate the issues around loop modeling. We actually started the project with doing significant amounts of loop modeling, but it just became intractable in terms of thinking about, how to really track that down. In terms of how much loop relaxation actually occurs in the design complex, we are eagerly trying to solve the structure of these complexes, and unfortunately haven't been able to get any structures yet, so I don't have an answer to that question. But the design intention was that there would be very little loop relaxation, and hopefully that will actually be borne out by the actual experimental structure determinations.
Thank you. I don't think there's another question, but I have one. Would this approach be generalizable to stabilize protein-protein interactions others than protein antibody, antigen-antibody interactions?
Absolutely, and there's been a lot of work in sort of the computational design area by many labs that have demonstrated you can use these approaches, these similar types of approaches, to optimize, stabilize protein-protein interactions. So absolutely.
Okay, let me see if there's another question. Yes, I don't think there is another question right now. So thank you again for this really interesting talk, Steve.
Thank you.